the last macromolecule that we're going to talk about today, but they're composed mostly of monomers or molecules, smaller molecules in a sequence with each other, repeating molecules that are sequential. So when we talked about carbohydrates, right? Carbohydrates are essentially this polymer made up of what monomer? What monomer were carbohydrates made up of? Go ahead.
Carbon?
Carbon is an atom that is a part of the monomer. Yes.
The carboxyl, right?
Carboxyl is a functional group. Yes.
Is it glycerine?
Or
Glycerol.
smaller subunits.
Glycerol. Sorry,
Not glycerol. Not glycerol. No.
Fatty acid?
We're talking. You guys are talking about lipids right now. Okay. You guys are talking about subunits of
Wait, I
lipids.
think I know it.
Subunits of carbohydrates.
Glycidic acid?
No.
Monosaccharides.
Smaller than that.
Saccharides.
Well, monosaccharides. Okay, so yes, so monosaccharides. All right, absolutely. So like glucose, right? We can have.
There it is.
So glucose, an individual sugar molecule. When that synthesizes, okay, we get these polymers. We call them carbohydrates. Okay, so that's big picture, right? Then we have proteins, right? Proteins synthesize. What are the monomers of a protein? Not necessarily how it looks, just the name of it. Yes, Joe.
The polypeptide.
Polypeptide is the kind of bond that occurs between these subunits called, yes?
Fatty acids?
No, that's lipids. We're talking about lipids.
Sorry.
Yes.
Like mentally stuck.
Amino acids, okay? So we have, those are two examples.
Good job, Jay.
Jay, those are two examples, okay, of polymers that are made up of biological monomers, okay? So then, when we're talking about lipids, we've already named the components of lipids, so I think we've got it, but they're not polymers in the sense of they're not made up of repeating monomers, okay? We have a glycerol backbone, right, which is that 3-carbon chain, okay? And then we have fatty acids that come off of that. So that's talking about fat as a very prevalent lipid, okay? So that would be the structure of that. So that's overview. Today we're going to talk about how nucleic acids, okay, so what are nucleic acids, and then what are the monomers of nucleic acids. So let's do that. First, before we do that, okay, this slide is saying nucleic acids store, transmit, and help express hereditary information. What does that mean? What does it mean that we're transmitting, storing, and expressing hereditary information? Greg.
It's like holding on like DNA and RNA to make another being.
Awesome. That's awesome that we're listing. We have a decent background knowledge that the two nucleic acids that we're going to talk about are DNA and RNA. Okay, those are the two nucleic acids. Okay, but my question is, what does it mean that we're going to transmit and express hereditary information? What does hereditary mean? Julia?
Doesn't it mean like genetic?
Genetic mean we got it from our
Parents.
parents, right? So from generation to generation, we are acquiring this information, this genetic information, in the form of nucleic acids in order to
Create dinosaurs, Jurassic Park.
Yeah!
or,
Let's go. Field trip.
World version
no.
2.0.
Or just synthesize nucleic acids. So
See, I can't booklet.
So we have DNA and RNA, all right, which we're gonna so big picture. Okay, these are polymers. We already understand, and the simple. I guess I'll just do a. Yeah, I'll do it like that. That's fine. But the simpler components of these polymers, we call those nucleotides. Nucleotides. All right, so these nucleotides, and it's for both DNA and RNA, so I'm just going to draw one line down, okay, branching from that. And the nucleotides are our monomers. And so we're going to, of course, monomers. And so we're going to talk about how they bond together and what they're structurally made up of. But essentially, big picture, that's what we're going to be talking about. It's going to happen in order to make up different structural components of us. So the DNA is providing the directions for its own replication. This slide assumes that we know a lot. So what I'm going to do is I'm going to give us like a very, very brief explanation of protein synthesis, because DNA, we say it unzips and then it becomes RNA, and it's this whole big process. And so I'm going to make sure that we have a really good foundation to now build up from. So we have DNA, okay, and that's in the form of this, what we call a double helix structure. Okay, so that's how you, in a two-dimensional sense, draw a very asymmetrical double helix. It's not usually asymmetrical, but that's what I did, and it's fine. So let's do some colors. All right. So we're gonna I'm gonna give our monomers just for the sake of
Differentiating.
Okay, I'm gonna give our colors and letters, and we're gonna call these our nucleic acids. All right. And so A is always gonna bond with T, and G is always gonna bond with C in our nucleotide sequencing. And this is what it is, but I don't want us to be too concerned with this at the moment. We'll get there. So we're gonna have this chain that wraps around. That looks terrible. I would like it to be in a line, and I'm gonna do it in kind of like
Ladder.
intervals of 3. Okay, I'm gonna do it in intervals of 3. So let's just stay right there. So if I know that this C is on this side, and I say that C always is put together with G, then I'm gonna have this and this and this, this. All right, and then I'm gonna need to fill in. I said I wanted to do intervals of 3. So here, 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 here. All right. And A bonds with T, so we have a complementary nucleotide across from it. All right.
Should we draw that?
If you want to. I mean, it depends on your understanding of protein synthesis. So if you've seen this before, no. If you haven't, here's what we're talking about. So DNA, we say that it unzips, okay, and that's triggered by mRNA, but RNA is the single strand of one of these strands. So we have 2 strands weaving together, okay? But we have to understand, like, this is the front, and then it goes and it wraps around and then it comes back. So we have this strand, and then we have this strand. All right, so we have 2 strands. We're going to pick this first strand, and we're going to look just at that strand. So we have red, red. We're going to have blue in the middle. Red, red, and then another red, another red. All right, great. Okay. So you guys see that I'm following this strand around and down, yeah?
Yeah.
Okay, so that's the strand of RNA that I'm looking at. And then I have a blue, and I have blue, blue. And I didn't realize that that's how I drew it. I kept them all on the same exact side. And it's okay. I wish I wouldn't have, but it's just going to be what it is. I apologize for that. I was trying to interweave a little bit better than I did, but that's fine. So this RNA is going to travel, all right, through, from the nucleus to a ribosome in a cell. All right, those are just organelles, and ribosomes are responsible for synthesizing proteins. All right, so protein synthesis occurs on ribosomes. That's great. And what occurs at the ribosome is that you have, and I'm just going to very, very simplistically draw what happens. All right, so these 3 nucleotides sequence for a particular amino acid. It sequences for that particular amino acid based on what it codes for. It's going to have codons, so there's anticodons and codons. That just means that it's coding for it, okay? And on that structure is going to be this opposing sequence. And this is an amino acid that was coded for because of the strand of RNA. And then the next one comes along and it's going to be bound to it because this next set of information in the sequence says this is what we need, right?